so let's talk about gene therapy in details now. Okay, so this is like a broken gene. This was something which was like it was supposed to be so hard, and it is becoming a piece of cake these days to do gene therapy. Something is missing, you just replace it. Okay, so if something is missing, you replace it, you add it. If something is broken, you fix it. Okay, if something needs to be deleted, you delete it. Everything is possible. Okay, so so let's look at, you can add an anti-angiogenic gene. You can stop angiogenesis for cancer. Okay, you can add suicide gene therapy. This is really interesting, suicide gene therapy. It is really interesting. CAR T cell therapy, immunotherapy, you can activate tumor suppressors. P53 is missing, add P53. P53 is defective, correct it, add it back. Okay, you can inhibit oncogene activation. Okay, so these are so many things you can do. You can do genome editing. You can, I taught a GYAN course in 2019, and two very young students came from far away place, like I don't know from where place, like really far away from Indore, and these were almost undergraduate students, these two students. That was a chemical biology course that I taught at that time. And they knew all about CRISPR-Cas9 in 2019. And they also knew all the backstory of CRISPR-Cas9, like, which I did not know, who developed it and who got the credit and kind of stuff. Okay, but it is still a very recent thing. So and that is an outstanding tool for genome editing. Okay, so I have listed 12 things here, but that is not all. That is just few of them. Okay, which comes under gene therapy. So this is really pretty interesting stuff. So I'll go through all of these 12 of them. Okay, so this is the picture. Okay, for all those 12 things. You can see there are some spelling mistakes here, right? Anytime you see spelling mistakes, you can safely say it was made by AI, because they can make it for you, but they cannot write this correct spelling. Okay, but they look so beautiful that I've still added it here. Okay. Okay, so let's look at gene addition or gene augmentation in cancer. So suppose you have to add P53 back, you use gene addition. So restore tumor suppressor, introduce therapeutic genes like immune activating or suicide genes. You can enhance sensitivity. So a functional gene is delivered into cancer cells or surrounding tissues using viral vectors, and this new gene supplements or replaces a defective gene and adds a new therapeutic function. So P53, P10 is also a very famous tumor suppressor. So that is deleted or defective in many cancers. So you can deliver a really good P10 gene. So here the difference is we are not adding the protein, we are fixing the DNA, or we are adding the DNA. This is not enzyme replacement therapy because we are not giving the protein, we are giving the DNA. So anytime there is gene therapy, we are talking about the DNA level therapy. Okay, that is the biggest thing. Okay, so tumor suppressor P53, this is approved in China actually, not elsewhere. So they restored P53 tumor suppressor in head and neck cancer, and this is approved in China. Okay, it is still not approved in US. Suicide gene therapy is a really, really powerful method. Okay, so you know, one of the things which is the biggest bottleneck in cancer therapy and therapy of many other diseases is that you can give the drug, but the drug itself is very toxic to the rest of the body, right? These drugs are not fun drugs because they are supposed to kill, and something supposed to kill is bad for normal cells also. So how can you hide the drug in such a way that it only gets activated in cancer cells? That is the trick. Okay, so what you do, you make a pro-drug. So this is a drug, but you add this pro part to that. This can go around anywhere in the body, but this is not active because it is pro. But once it gets to the tumor cells, then only this thing is removed. It gets into the tumor cell and they die. So that is suicide gene. So this is a very famous approach and pretty well established approach. So you insert genes that convert a non-toxic prodrug into a toxic compound in cancer cells. So we don't have this gene in our body. Nobody has, no human has this thing. But what we do, if somebody has cancer, you have to deliver this DNA to that person, okay, in the tumor cells. So this DNA is for HSV-TK gene, okay, and this is a simple DNA, and all it does is that it adds a phosphate group here. So it's not a bad DNA. It is like a kinase which is adding a phosphate group. It's as simple as that. Okay, so you add this thing, and the substrate is this one here, ganciclovir. Okay, so you add this drug ganciclovir and this gene there, and this gene is expressed only in the tumor cells. So what happens is that this drug is floating around in the whole body, nothing happens, right? But in the tumor cells you have delivered this DNA, so this DNA will make this protein and it will phosphorylate it. So once it becomes monophosphate, then other kinases in the body will make the corresponding triphosphate. This gets, see, this is very similar. It does not have the sugar ring, but it is very similar to nucleotide. Now because it does not have the sugar ring, once it incorporates, the 3' hydroxyl group is missing. So the chain cannot continue, right? It is the phosphodiester linkage, 5', 3', right? 5' is there, 3' is missing. So it will get incorporated, but nothing can happen. So DNA chain will stop there and the cells will die. Are you guys following that? All of you? Because it's a 3', 5', 3 to, you know. Okay. So that stops here. So what you do is that you can give the suicide gene exactly to the tumor site, then you deliver the drug, okay? And then it will only work in the tumor thing. This is suicide gene therapy, okay? So look at this thing. You deliver the drug, the protein is made, the prodrug is only converted in the tumor cells, not in the rest of the body, and the tumor cells die. Now these drugs have a very interesting effect also. When tumor cells die, what happens is that they release this drug in the media, and this drug then goes to the actual triphosphate, just gets into other cells, healthy other tumor cells, and they also die. It's like a bystander effect. Once the tumor cell slides and the drug is released, it just goes and goes to the neighboring cells, and they all die. Okay. So that was suicide gene therapy. Now let's look at immunotherapy with gene modification. So we modify the genes to get immune response. Okay, so I will tell you about 3 things here. Deliver genes encoding immune activating cytokines, cancer vaccine, and dendritic cell therapy. So if you add the genes which can make these cytokines, so once these cytokines are released, these cells come close by and they will eat up the tumor cells. So in tumor cells, you give the DNA so that they make these cytokines, and these cytokines, when they are released, this is a signal to attract the immune cells. What do macrophages do? They eat up the cancer cells. Anything comes in close contact, T cells, they will have a strong immune response, NK cells, so they get activated. So you can directly deliver, actually my mistake here, you don't even have to deliver the DNA for the enzymes which make it. You can actually deliver the DNA for these things directly, and they will be synthesized and released directly. So that's even simpler than that, right? You can make IL-2 in the lab. You can take the DNA, put it in E. coli, it will make it for you. But here we are putting it in we are delivering it to the tumor cells, and once it is released, these macrophages will come. Cancer vaccine therapy, you already know about it, right? You know the vaccine thing. So you take irradiated, so in this is pretty interesting. You take GM-CSF secreting. So GM-CSF, when you take tumor cells which naturally excrete GM-CSF, or you can engineer it so that they secrete it. Now, why do you take tumor cells? Because you want to start an immune response, and then you irradiate it so that it can no longer divide, but it has all the antigens there, right? You add it there, and you inject it in a patient. So now you have an immune response, but you also have GM-CSF there. So GM-CSF is like a signal to attract all these immune cells there. So immune cells will come at the place where these tumor cells are there, and they will have a strong immune response. So DC cells uptake tumor antigens from irradiated tumor cells, and once they take it, then there is an antigen presentation. T cells will be activated, B cells will make antibodies, and there will be a strong response. Okay, even if it is not releasing GM-CSF, it's okay. Anytime you give a vaccine, immune response is going to be turned on. Okay, that is the whole thing. Dendritic cell gene therapy. So you engineer dendritic cells so that they release cytokines. Okay, cytokines, when they are released, they will attract macrophages and other immune cells. Okay, see, keep in mind what you want to do here. If you want to boost an immune response, it is very simple. You have to call the immune cells close to the tumor site. How do you call the immune cells? By releasing cytokines. Cytokines are if you release the cytokines, the immune cells will come running in that area. Okay, so if you want to boost and you have to release cytokines, how do you release cytokines? You put the DNA for the cytokines there. So cytokines will be synthesized, they will be released. When they are released, immune cells will come close by, they will eat it up, or they will kill it, right? So that's immune boosting. Or you can deliver the drug, or you can take NK cells, which are immune cells, and you also make them like that they are releasing cytokines. So they will go there, they will release cytokines, more immune cells will come.
While we were discussing about nanotechnology, you discussed about folate with drugs.
That is not even nanotechnology. Folate, you don't even have to do nanotechnology packaging or something. You can, it will directly bind to the drug. You can give it in the bloodstream, it will bind to the drug. The folate receptor at the surface of the cancer cells. Okay, so dendritic cell therapy, you take dendritic cells, you add the gene for the antigen and the cytokine. What antigen? So you know tumor cells express this protein like a spike protein. They don't, but let's say spike protein. So you take the DNA for the spike protein, you put it in NK cells. You put cytokine genes also in NK cells, right? Now these NK cells will express those spike things, and they will also express cytokines. Now they are releasing cytokines. Once they are inside, immune cells are coming. Now the spike is there, now the immune system is going crazy. So tumors, once they make the antibodies, tumor cells actually have those spikes, tumor cells will be killed. Okay, so antigen presentation, cytokine secretion, huge immune response. Okay, so that was immunotherapy with gene thing. Oncolytic virus therapy, I already told you before. I am going to tell you here also. So there are three mechanisms by which oncolytic viruses work. So oncolytic viruses are viruses which cannot divide in normal cells and which will not infect normal cells. They will only go in cancer cells first thing. They will only replicate in cancer cells. So let me repeat what viruses do. Even if one virus molecule infects a cell, it will go inside and it will replicate, right? Replicate, replicate, replicate. And it's like a bag full of viruses there, trillions of viruses. So the cells will, what will happen to the cell? It will burst open. All the viruses will be released. So the cell is lysed, right? These viruses will infect fresh cells, make a bag of cells, they will burst open, and they will just keep on amplifying till the whole thing is gone, right? So that's what oncolytic viruses do. Even if you don't modify them, all you have to do, they don't touch normal cells. They go in tumor cells, they replicate in tumor cells, and then they will lyse the whole tumor. It's all gone. So that's one way. So that is direct lysis. When you lyse it, what will happen if tumor cells lyse? All the antigens are showing up. So immune system will be activated. It has to get activated, right? Third thing is that you can, inside the virus, you can add toxic thing. So those things will further kill the tumor cells, right? So that's what is.
These viruses, even by accident, will not infect any healthy cell.
So they are made it in. So this is known for quite a while. You can make replication defective viruses which will not touch normal cells. So in our lab, we work with viruses all the time. If we even touch it, it will not affect us. Okay, we will not touch it, but they cannot do that. So oncolytic virus will not touch normal cells, malignant cells, and they will lyse it up. Because they lyse it up, all these immune cells will show up and a huge immune response will start up. Okay, and once the immune response starts, it will clean up the whole mess. Or you can, inside the oncolytic virus, you can put some very toxic thing which will be expressed, and that will further kill the tumor cells. So there are three ways oncolytic viruses work. Then you can add genes which will inhibit blood vessel formation. This is anti-angiogenesis thing. You know, anytime somebody is teaching, somebody is treating cancer, aggressive cancer, anti-angiogenesis inhibitor is always part of the therapy. Stopping the blood vessels is always part of a therapy. Okay, so what you do, you can add anti-angiogenic, inhibit blood formation, blood vessel formation by introducing genes that produce anti-angiogenic proteins. So one example is endostatin gene, which will inhibit VEGF is the ligand for angiogenic pathway. VEGF is vascular endothelial growth factor, which binds VEGF receptor, and that is where the signaling is turned on. Okay, so if you add endostatin gene, basically it inhibits angiogenesis. So if you have to add a gene, add this gene, and this will prevent blood vessel formation. Okay, then gene therapy for drug sensitization. So you want to make tumor cells more sensitive to the drug so that normal cells are spared and tumor cells die. So one of the things they do is to add cytosine deaminase genes, which converts 5-fluorocytosine to 5-fluorouracil, which is the active chemotherapy drug. So if you put this only in tumor cells, only tumor cells will make it, right? Normal cells will not make it. So this drug will float around in the bloodstream with no impact on normal cells, only tumor cells will die. I can see you guys are very tired. This is the fourth one. But some more time to go, and then we'll be done, at least the lectures will be done. Okay, so you give it only to the tumor cells, and this 5-fluorouracil will only be formed here. Okay, so 5-fluorouracil is toxic in many different ways. So 5-fluorouracil creates oxidative stress, it inhibits DNA synthesis, it causes replication stress. You know, the replication, when the DNA is doing it, it is how many of you have studied DNA replication in the course? Most of you. You know how many proteins come together for DNA to open up and replication to occur. So when you add these kind of compounds which mimic the bases but they are not the bases, there is a huge amount of replication stress. Okay, and that basically kills the cells. RNA gets, uracil is part of the RNA thing. So RNA is also affected. So all DNA fragmentation occurs. So all of these things basically kill the tumor cells. Okay, so these are different kinds of vectors which are used to deliver these DNAs, adenoviral, lentiviral. Sometimes you can take plasmid DNA, and sometimes you can package them in nanoparticles also to deliver the DNA. Yeah, go ahead. You have to speak with that thing. Who has it?
Ma'am, as you said, how is 5-fluorouracil is selective to cancerous cell?
5-fluorouracil? One way is that, so thymidylate kinase, thymidylate synthase enzyme, the role of that is to add the CH3 group to make a thymine, right? So 5-fluorouracil is very similar to thymine, so it can bind thymidylate synthase enzyme and inhibit it. So that's one way. Second thing is that 5-fluorouracil can get the sugar with the phosphate can get attached to it because it's very similar to thymine, and then it is converted to di and triphosphate and then get incorporated in RNA and DNA. And if gets incorporated in DNA and RNA, it's going to damage the DNA and RNA, right? Because it's not the natural base. So then, DNA repair enzymes may be able to fix it, may not be able to fix it. But the DNA structure is damaged. RNAs are damaged. So all of these things, then 5-fluorouracil will kink the DNA structure, will, distort the DNA structure because it's not the natural base. So anytime something else comes in, it is going to distort it. So, and when it is getting replicated, it will create replication stress. 500 to 1000 bases are added per second, so it will create that. So replication stress will turn on warning signal. So it just keeps adding up and the cells will die. Make sense? All right. Gene editing, cystic fibrosis, yes.
How do you make it specific to the cancer cells?
Oh, so that is what was written here actually. So you do not give 5-fluorouracil as such, because if you give 5-fluorouracil as such, it will go to all the cells in the body, and so you are giving it as a prodrug, and like in this form, but you are delivering this enzyme specifically to tumor cells. This enzyme, if it is present in every cell will make the same thing. So you don't want to do that. You want to give the uniqueness to tumor cells. So you are adding, so remember we're talking about gene therapy now. So we are adding this gene, so we are delivering not the protein but the DNA of cytosine deaminase to tumor cells. So once that protein is made, then this drug, this product, cytosine deaminase will be converted to the 5-fluorouracil. R is fluoro group here. Okay, and once it is there, so this is present only in cancer cells now, right? And then it will kill that. You don't want to do this as a systemic therapy. You don't want to give 5-fluorouracil. I mean, it is given as a chemotherapy, honestly, but it is very toxic. Chemotherapy relies on a very simple principle: kill the dividing cells. Cancer cells are dividing faster than normal dividing cells, so it's just but it is a pretty bad thing. But even today, chemotherapy is given to the people who have the best healthcare access in the world, because sometimes you have to give it, right? So you guys heard of Kate Middleton, right? Princess of Wales, right? She had cancer and she was given chemotherapy also. And she obviously has access to the best things. So chemotherapy is still widely used, depending on. But if it is cured fast enough and the person is young enough, it's good. Old people, it's very hard. Older people, it is very hard. Gene editing. Okay, so if there is a defective gene like in cystic fibrosis, you can use many different ways. CRISPR-Cas9 is obviously the best these days, but TALEN and ZFN method is also extremely useful. To correct it, then put it back in there. Okay, so the person I was telling you about, whose son has cystic fibrosis gene, so she was telling me that she's hoping that by the time his son grows up, the science is there to correct it, and it is there to correct it now. Okay, so now let's look at the case study, and this is where it gets very interesting. We all know about sickle cell anemia. So normal red blood cells look like this. Sickle cell anemia, they look like this. And there is only one mutation in the hemoglobin gene which makes it like this. So what did these red blood cells, when they are donut like that, they are very flexible. They can squeeze in and very nicely they can go. They have a half-life of 120 days. But this one single mutation makes them sickle cell-like. They die in 10 to 20 days. and then they aggregate and they block the blood vessels. So it's a very bad disease. So this can be fixed by CRISPR-Cas9 or gene editing techniques, and you can put it back. Okay, then you can, many times P53 is not deleted, it is just mutated. So you can fix the mutation, you can fix the DNA, put the correct DNA back in the body. Okay, same thing with BRCA1. APC is also tumor suppressor, you can do the same. You can actually knock out DNA repair genes in the tumor so that if you give radiation, they die, right? You make them very sensitive because no DNA repair genes, chemotherapy, irradiation will completely kill them. Okay, multi-drug resistance occurs because this ABCB1, this PGP glycoproteins pump it out. You remove the gene, now they cannot pump it out. So that will work. Okay, so CRISPR-Cas9 technology has made huge. You know, we were making a mouse. I was collaborating with Paul Greengard, who was the Nobel laureate in 2000 or 2001. He was at Rockefeller. So we were both working on CDK5 kinase. So we were collaborating, and so he said he was making the mouse, genetically engineered mouse with CDK5 all the time. So he said he will make the mouse, and we divided the labor. So he will do these experiments, I will do this experiment. So in those days, it used to take almost 5 to 6 years to make a genetically engineered mouse. In our case, it went up to 10 years. And even after 10 years, the mouse wasn't ready. Okay, it was that hard. Okay, and if you make an engineered mouse, you would definitely get a Nature paper because it was such a big deal. CRISPR-Cas9, you make a mouse every 6 months or even shorter period of that. It's like a real piece of cake. It's such a huge advantage. Okay, so the kind of things you can do with CRISPR-Cas9 is just enormous. Different things you can do. You can edit any gene, you can put it, you can change it, you can. So if you want to make superhuman one day, this is the easiest way to do it like that. Okay, you can, any mutation which makes them bad, you fix it. You just completely fix it. Okay, and they become normal. Okay, gene silences and knockdown. So how many of you have heard of RNAi? Only you. You other people, you have also used, but most of you have not heard of RNAi. So RNAi was a method which was discovered in 1990s. So what they did is that these scientists who were working with plants, they were not even working with, you know, animal stuff. So this plant was purple in color, so they put double the gene to this purple plant, thinking this will become dark purple. Instead, it became white. Okay, so when you add more gene, you would make it more darker than this became white, and that led to the discovery of RNAi. And that time they thought it was only in plants, but it happens in humans also. So basically, when you have the messenger RNA and you put a small piece of complementary messenger RNA, complementary, so that binds there and the cellular machinery kicks off and degrades the messenger RNA. So what happens? Protein is not expressed. Okay, so suppose I want to degrade a protein. So DNA makes RNA, RNA makes protein. So I degrade the RNA. So if RNA is not there, protein will never be made. That is called RNAi or RNA interference. So that became so popular that was discovered in 1990s, but that was a game changer. Okay, that time obviously CRISPR did not exist, so all of us were using RNAi to knock down genes. Okay, with CRISPR it is a piece of cake. You can knock out gene. You don't even have a trace of that DNA. It will never make it. Go ahead.
Can we engineer RNA also?
Please, you can engineer RNA all the time. CRISPR works on DNA. Yeah, because if you do the DNA, it is a permanent thing, right? RNA will always be based on the DNA. Yeah, it has a shelf life. But RNAi is extremely, like for last 25 years, people are using RNAi so extensively. Even today, it's just so easy to use. Okay? So that's what it is doing. If you want to silence a gene, you can use RNAi or you can use CRISPR. So if you use RNAi, see, knockdown and knockout. Knockdown means you are reducing the level of it, knockout means complete removal. So cancer cells often rely on overexpression of oncogenes, angiogenic signals. So if you put the RNA, corresponding RNA for these things, they will be degraded. The cancer will disappear 100%. I mean, tremendously. If you are doing RNAi against the main oncogenic driver. So if you put the RNAi against MYC, complete reversal of cancer. So it makes gene silencing, right? So it inhibits tumor growth, it enhances sensitivity to therapies, okay? So these are some of the things, gene silencing targets, BCL2, KRAS, oh my God, so important. VEGF, this is angiogenesis one. If you make it disappear, make EGFR, PDL1, they are not even expressed. So the point is you are attacking at the RNA level, right? But now you do it with the CRISPR, you attack at the DNA level, and that is permanent, okay? So this is current clinical trial and successes. This is about the so you can make PD-1 knockout. Knockout means you have used CRISPR in this one. See CRISPR Cas9 here. You can do many different things. All right. Now what are the challenges in cancer gene editing? Off-target effects. You want to cut only at one place in the whole genome, but if the sequence is a little bit similar, it can cut at the wrong place too. So you have to be very smart about it. Okay, tumor heterogeneity, it's not only one kind of thing. A tumor has many different kind of things going on. So if you target one gene, you may not be able to reduce the tumor completely because there are other factors also going on in there. Okay. It is hard to target deep or solid tumors directly, especially if they are deep inside the body. Skin cancer is easier to treat because it is outside. Surgery is possible, therapy is possible. Something which is deep inside is harder, especially the tumor is very deep. And obviously the ethics and regulatory things always kick in. Okay. Mechanisms of gene silencing RNAi. Before RNAi was discovered, people were using antisense technology, oligonucleotides. Do you guys know about antisense thing?
5 times, 3 times the opposite complementary.
Complementary sequences to do that. Yeah, there was a big company there. This company was close to San Diego in a place called Carlsbad, which is really beautiful in California. And the company's, it was a very famous company. The company's name was ISIS. And when ISIS, the other ISIS came into existence, they had to change the name of the company because it was like nobody would want to even hear the name ISIS there. So they were the world leaders in antisense oligonucleotides. Okay. MicroRNAs or mimics or inhibitor, CRISPR. Without going into details, these are the ways you can knock down the gene. CRISPR will knock out the gene. Okay. So siRNA, 3' are small interfering RNAs. shRNA are the same thing, but instead of giving RNA every single time to the cell, you clone the DNA in a vector, in a plasmid, and you give the plasmid to the cell. So this will make the RNA all the time to complement with the messenger RNA to degrade it. So once you do it, you don't have to worry about it anytime. It will always keep the expression down. Okay, but if you use siRNA, you have to keep adding it every few days. Okay, because RNA will degrade, then you add it again, then you add it again, then you add it again. Here it will constant supply. Okay, antisense are chemically modified DNA or RNA sequences that bind complementary messenger mRNA. Now nobody uses ASO because we have so many better techniques. Okay, CRISPR is there. MicroRNAs are also small pieces of RNA which will bind to the sequence, and when they bind to the sequence, they can degrade it, or they can bind to the promoter region and inhibit transcription, or they can bind to some part of the RNA and they can inhibit translation, or they can activate translation. So they have a variety of functions. Okay. So what are the advantages and limitations of gene editing? You can do highly specific targeting, but delivery challenges are there. Reversible in most cases, off-target effects are possible. You can target undruggable. If you're targeting DNA, there is nothing undruggable about DNA. DNA is just a sequence. You can definitely target it. So these kind of things are often used in combination with other therapies. Okay, I can see you guys are extremely tired. Maybe 4 lectures is not a great idea. Think about the Christmas thing once again. Okay, this is just a beautiful picture. It's not telling you much. Let's go to CAR T therapy. So I will tell you about a CAR T therapy, but just extremely briefly, because we have 2 lectures devoted on CAR T therapy, and there I'll show you all the structures and how they work. But basically CAR T cells, so T cells, so we take the T cells out of a patient or from a healthy donor, and then we engineer CAR. And what is CAR? CAR is chimeric antigen receptor, CAR. So what they do, these receptors, they bind to tumor antigen and they turn on the immune response. So CAR T cells are equipped with all the things there, and there are many varieties of CAR T cells. So we are here in this point. So you will see there's a lot of overlap between one point to the other. As I've been telling you 12 different ways, you will find overlap. So because it is an immune thing, I have included PD-1 knockout, but I also included PD-1 knockout in the gene knockout thing. So it doesn't matter. We have classified it, but I'm just telling you all different ways here. Okay, so one thing is that you can remove immune checkpoints. So do you guys remember about the PD-1, PD-L1? Can anyone explain to me what exactly is happening? Because this is very important. So normal cells, many cells, APC cells, antigen presenting cells, they express PD-L1, which is, L stands for ligand, and T cells, they express PD-1. So if PD-L1 locks with PD-1, T cells become quiescent. So this is a natural mechanism to inhibit autoimmunity. So T cells, when they're hyperactive, they will start destroying everything, and in that hyperactive state, while they are cleaning up all the foreign mess, they can also damage our body. So that is why this is why nature has designed the PD-L1 on APC cells and some other cells. They come and they lock with T cells, and T cells become quiescent. So what tumor cells do, they also express PD-L1 and they lock with T cells, and T cells become quiescent, right? So if you break this interaction, so how do you break this interaction? You make a PD-L1 antibody which has even higher affinity. So now tumor cells have PD-1 and T cells have tumor cells have PD-L1 because they are locking it with this one. So T cells have PD-1, and if you make PD-1 antibody, that will be locked, so tumor cells are going to be there. Or you can make PD-L1 antibody, so you can lock the PD-L1 on tumor side so that they cannot form that bond with the T cells. Is it making sense? Right? So you can even remove PD-1 completely from tumor cells, right? PD-1 from T cells. So if you remove T cells, they don't have PD-1, tumor cells can express PD-L1, but there is nothing to bind there. So you are making T cells which cannot be switched off, right? Because there is no PD-1 there. Not making sense? No, I will show you the picture, because this is something you have to show it via pictures. This is too much information. So T cells have PD-1, okay? T cells have PD-1. tumor cells are not supposed to have the ligand for PD-1, which is PD-L1, but they do express it. So this PD-L1 locks with PD-1, and as soon as it locks, these cells become quiescent. Okay. So what if you remove PD-1 from T cells? Okay. Now T cells are going to be active all the time. So PD-L1 has nothing to bind. So they will remain active and they will kill cancer cell. Is it making sense? So that's what it is. So that is what was said there, that you can engineer T cells to have no PD-1. And if they have no PD-1, there is no off switch there. You keep them on all the time. So they will kill the tumor cells. Now let's go to CAR T therapy. So what you do, you take out the T cells, which can be from a healthy donor, allogenic, or you can take it out from the patient itself. And then you grow it in the lab, and then you add the DNA through a virus into these T cells so that they express these receptors. These receptors are these are the natural receptors. When you put the virus in there, depending on the DNA, now they will make these receptors. These are the chimeric antigen receptor, these orange ones. Okay. So what are these receptors binding? They are against the cancer cells. So when cancer cells come, they bind, and once they bind, they basically kill the cancer cells. They also release cytokines. There are many different kinds of CAR T receptors. Okay. So cancer cells eventually dies. That's CAR T receptor. Okay. Here is the thing. Suppose a patient has cancer and you take out T cells from them. The patient has probably undergone chemotherapy or radiation therapy. So the patient is not healthy at that stage, and you are taking out T cells from the body, which is probably the only way to do it, but it is even more taxing. Then you engineer all these things, which may take weeks to months. The patient may not have that long to wait. So in ideal situation, you want CAR T cells ready so that you can directly inject it in the patients. So what they do, they take it out from healthy donors. Okay. So autologous means that you are taking it from the patient and putting it back in the patient. So there is no immune rejection here. But allogenic means you are taking it from healthy donors, and there is going to be immune rejection, right? Especially if you give to many people, high percentage of people are going to reject it. So the good news is that by this time we already know what are the features in these receptors, like what are the warning signs for the body to recognize it as self versus foreign. So what we do, we know the body is going to look for these 5 receptors as self versus foreign. So we remove those 5 receptors. Okay. If we remove it, immune system cannot recognize whether it is self or foreign. Make sense? So this is what is shown here. You don't have to remember anything, but see. So there are 2 things which can happen. The donor T cells attack host tissues because donor T cells recognize host as foreign. So they are attacking. That is called graft versus host disease. Forget about host attacking T cells. The foreign T cells are attacking the host. So we know what leads to that. So T cell attack via T cell receptor, TCR. Remove TCR, T cells cannot attack the host cells, right? So we can fix that problem. But then host cells also will attack the T cells as foreigns. So we have to remove all the things which will allow the host cells to recognize T cells as foreign. So in here, this B, C, D, E, E. So in this one, we have done TCR knockdown, T cell receptor knockout. So when we do that, this cannot happen. So T cells cannot attack the host cells. So we have taken care of one problem. Now we also want to make sure the body does not attack the T cells, otherwise they will be gone. So there is so many things we have to do. We have to knock out CD52, we have to knock out CD7, we have to change HLA A and B. Then we have to do a lot of things. We have to knock out HLA2. These are different kinds of modifications we have to make before we add the T cells, and then the host body cannot recognize that this is foreign. All the things by which they recognize have been changed or removed completely. So human body cannot recognize this as foreign either, right? So now the CAR T cells are ready in the pharmacy, right? The moment you need, you can add it. Patient is not going to reject it. They are not going to attack the patient, and they're ready to go. So the doctor writes it, you immediately get it. Okay? So that is prevent rejection in allogenic off-the-shelf CAR T cells. So you engineer CAR T. So they are showing all the receptors, chimeric antigen receptor, but they also have all these other features there. This has already been done. Okay, we already know exactly how to do it. So let me ask you, what are these CARs? What are these CARs against? What is the function of CAR, chimeric antigen receptor?
Recognizes the antigen specific to the
Tumor cells, very good. So even if you give in the bloodstream, it is going to only go to the tumor site and get locked there and kill it. Okay. So that was CAR T cell therapy in very simple words. Then gene blocking therapy is very simple. So this is a gene which is, overexpressed. You add a blocking gene, it inhibits the expression of this thing, cell works normally. You take a cancer cell, you make it normal. Okay, you don't even have to kill it. Then let's look at ex vivo versus in vivo. Do you know what is ex vivo versus in vivo gene therapy? Can anyone explain to me? Yes. You need that. Anshul, right? Anshul? Rohit. I should not even try to say the names because I'm going to use all the wrong names on record.
Like ex vivo gene therapy suggests we are taking out the cell from the body and we are correcting it, and then we are adding it back again. And in vivo means it's on the inside the
body
host.
It is being corrected. So there are two ways to correct it. That's exactly what it is. Okay. So in ex vivo, you take out the cells from the body, you modify it, you put it back in. For in vivo, you do the gene therapy directly in the human body and it is corrected there. So ex vivo, you collect it, you change it, and then you turn it back there and you constitute it. Okay. And in vivo, you directly give the local injection to the site and then it corrects itself inside the body. Okay. The last thing is epigenetic gene therapy. Okay, why epigenetic gene therapy? Because epigenetics is the one which is controlling gene expression. So you can actually do epigenetic gene therapy. So for example, this TET1 is a demethylase. Okay, so this is switched off. You can give the DNA to this DNA, the gene is turned on. Okay. DNMTA3A, it is turned off. You can give the, this is hyperactive and it is doing the tumor suppressor methylation. So you can turn it off by knocking it out basically through Cas9. The gene is turned off. Similarly, you can do for P300, you can add histone acetylation so that it opens up and it expresses P300 protein. For KRAB, this is an oncogene, so you add repressive histone markers. What are repressive histone marks? So that which will switch off gene expression. Which one? Methylation. Very good. Methylation of histones. So this is highly precise and modifies gene expression without cutting the DNA. Okay. So I'm sorry, here when you do it, here you add repressive histone marks, you do not, for this you do target methylation, you don't knock out because I'm talking about epigenetic markers here, okay. So this you already know, right, epigenetics. So, and so one thing is that if you have to use it through small molecule, you use the inhibitor. If you use epigenetic way, you can do the gene expression by changing the epigenetics there. Okay, and that's it. We finished it.